Hello and welcome to BioRules video lessons. Today we are going to be talking about the angiosperms life cycles. So the life cycle, it's practically the same as what we saw before. There will be some small tiny differences. So the first thing, that the plant is still the sporophyte, is still diploid. So let's take a look on the micro part of the life cycle, the male part of the life cycle. So if you take a look on there, we have a microsporangium, this place that's going to be producing the spores. It's the same as it was before, and by meiosis we are going to be making many spores. The spores are going to be dividing and making a pollen grain, which is the microgametophyte. It's the tiny little plant, the equivalent of the heart in the pteridophytes, the equivalent of the little plants in the bryophytes. It's exactly the same as it was before in the coniferophytes. The pollen grain is the microgametophytes. The microgametophyte then is going to be having the generative cell and is going to be having a tube cell. The tube cell grows and is going to be reaching the female part. The generative cell is going to be dividing and making the sperm cells, exactly as it was before for the coniferophyte. So it doesn't change anything. We have here the generative cell giving rise to the sperm cells. We have there the tube cell giving rise to the tube, and the tube is going to be growing and it's going to be reaching the female part. Exactly the same. So what's going to be different? The difference will be in the megaparts. So we have a megaspore mother cell, the cell that will make the megaspores by meiosis. So we have one megaspore, the other three are going to be dying, and now we are going to have mitosis, but different from the coniferophytes that we have many mitosis here, making the oosphere and all the endosperm over there. We are going to have only 3 mitoses. So that's what's going to be having here. So let's take a look on that. So that's the megasporocyte, the cell that gives rise to the megaspore. It's going to be suffering meiosis. It's going to be making 4 cells. One of them is called functional megaspore, which is already haploid, suffered meiosis. 3 of them are going to degenerate. So we have 1 mitosis, makes 2 cells. 2 mitosis, 4 cells. 3rd mitosis, we end up having 8 cells. If we organize those cells, they are going to be looking like this. We have the antipodes, we don't care much about them. We are going to have the oosphere, very important. It's going to be the gamete of the females. We are going to be having there the synergids. They stay beside the oosphere, they help, they give support to the oosphere, but not that important as well. And we have here, very important, the polar nuclei. The polar nuclei is going to be very important as well. So the 2 most important here, oosphere and the polar nuclei. So if we take a look on there, we have the antipodes, whatever. We have the oosphere, very important, the synergids giving support, and we have the polar bodies. This is the structure of what would be the megagametophytes, waiting to be fecundated by sexual reproduction. So how is this fecundation going to happen? The structures over there, and the pollen grain hits the top of the flower into the little structure called the stigma. The pollen tube grows to the style and reaches an egg. So here you have the pollen tube growing and it reaches the oosphere. And inside this pollen tube there will be 2 sperm cells. One of them is going to be fecundating the oosphere and is going to be making the zygote, which is diploid, which will grow into the embryo. However, in angiosperms we have a double fecundation. The polar bodies is also going to be fecundated by the other sperm cell. So now we have polar body, haploid, another polar body, haploid, a sperm cell, haploid. The 3 of them are going to be making a cell that's triploid. Yeah, that's weird, that's different. So this triploid cell is going to be dividing, and is going to be making the endosperm, the nutritive part of the seed, which in the case of the angiosperms, it is triploid. Okay? So, putting things together here, this is the endosperm growing. It grows, and the embryo also grows, and the seed grows even more, and the embryo grows over there. So that orange part over there is going to be called the endosperm, the nutrition, which is triploid, and we are going to be having the embryo, which is diploid. This is going to be making the angiosperm seeds. Okay, so shall we compare conifers and angios seeds? So let's make it side by side here. On the left we have the conifers, on the right we have the angios, and they all start with a megaspore, which is already haploid in both of them. So this megaspore is going to be doing many divisions on the conifers, and many divisions there, which is going to be making all the nutritive tissue, the endosperm, haploids, and also going to be making an oosphere, which is also haploid. This is what happens in the conifers. What about in the angiosperms? 3 divisions, so it's going to be making all those cells that you have seen before. You are going to have the oosphere, which is also haploid, but we are going now to have the polar bodies. Each of them is also haploid. And here comes the male microgametophyte, the pollen grain. In the conifers, they hit the bottom of the seed over there. In the angiosperms, they're going to be hitting the top of the female part of the stigma. And then, both of them, they are haploids, and they have a tube cell and they have a generative cell inside. Both of them exactly the same. The tube cell grows. In the conifers, it grows straight to the oosphere, and in the angiosperms, it's going to be growing through the stigma, through the style, get into the ovary, and inside of them we are going to be having sperm cells. On the conifers, the sperm cells, which are haploid, are going to be fecundating the oosphere, the same as will happen into the angiosperms, and they are going to be making the zygotes, which in both cases is going to be a diploid tissue. The difference now is that, this is not different, this is the same. Both of them are going to be growing an embryo. You're going to have an embryo grown from the zygote in the conifers and the angiosperms, but the difference now is that the angiosperms have a double fecundation. The second sperm cell is going to be fecundating the polar bodies and is going to be making a triploid endosperm, which is going to be growing, is going to be making the nutritive tissue of the angiosperm seed. And we are going to have there a triploid tissue. So that's one big difference. If you eat a pinhão, the conifer seed, the pinhole as well, you are going to be having a nutritive tissue which is haploid. So we are going to have this here, is going to be haploid. But if you eat beans or rice or any other flowering plant seeds, the nutritive tissue is going to be triploid. So yes, when you're eating beans, you are eating the embryo inside, okay, that's diploid, but the nutritive part is going to be the triploid endosperm. So here we have a few diagrams for you to get familiar with, if you want to see later. But what if we want to put the angiospermatophyte's life cycle into the square diagrams that we had before? So, sporophyte, diploid, is the plant over there that produces flowers, and is going to be having the male side and the female side, and you're going to be having the meiosis happening in the anterior or inside the ovary. The meiosis is going to be producing the spore, which stays inside the sporophytes. And then, of course, if it was the male part, it will make a microgametophyte, which is the pollen grain. The microgametophyte will have a sperm cell in it, which is haploid as well, while the female part will make the megagametophyte, which in the future is going to be the seed. And this is going to have a noosphere, the gamete, which is also haploid. Now, the pollen grain usually taken by animals, there are exceptions, but the most common is that the pollen is taken by animals to the female megagametophytes, what will be the seeds to the flower and then get to the megametophytes, and you have the fecundation. The fecundation is going to be making the zygotes, which now is diploids, and that will be inside the female gametophytes. This will develop into the embryo inside the female gametophytes. Not to forget, there is a second division, okay, into the seed over there. In addition to what we had, we have the two polar bodies down there in yellow, and these polar bodies are going to be fecundated by another sperm cell. And when it fecundates, it makes the endosperm, which is triploid. Okay, and the embryo is still there. It's that green part in the middle. So this here is the embryo. There it is. Okay? And now it's time to exercise. If you read this QR code with your cell phone, or if you enter in the link that's below it, you're going to see a questionnaire with a few questions for you to do in the end of this video. Okay, have fun.